Bonjour tout le monde. Bienvenue à Paris. I'm Anna-Marie Wagner. It is absolutely wonderful to be here. I did iGEM the same year many of you were born, I found out today, which was great. And I was walking through the little kind of wall of history down there. If you look at I think it's called Food and Nutrition now. It used to be called Food and Energy. The 2008 iGEM team from Harvard won that, so woohoo. You all have done a lot more than we ever accomplished in our project. It's been inspiring and humbling to walk through the various sections this morning. So first of all, just congratulations to all of you for the work you've done over the past year and for being here and for advancing this field. I'm really, really, really proud to see what this community has become over the last over 15 years. This is going to be a panel about AI and AI's impact on synthetic biology and synthetic biology's impact on AI, which I'm actually much more interested in and excited about. I really don't want this to be just another panel about, is AI hype or is it real? And so we're going to try to get a little bit deeper. And one of the topics that I wanted to maybe just start with some reflections on is the culture of synthetic biology and the culture of computer science and AI. And I think this community in particular was really inspired in many of the early days by the computer science community, by the sort of engineerability of computer science, and by the potential to treat biology like an engineering discipline, right? A, C, T, and G, it's just like zeros and ones. If we can read it, we can write it, we can program it. I have been guilty of saying that many, many, many times. And while to some degree that's true, we all know that you can program something one day and program it the next, and it doesn't run the same way in your machine the way that if a computer scientist programs code on their machine, it runs the same way every time, predictably. Biology is complex. The machines are complex. We did not invent it, right? We like to say biology invented us. And so we're students of this incredibly complex machine. And so there's been this sort of tension between these cultures, this culture of computer science and engineerability and control and predictability, and the culture and reality of biology, which is sometimes random, and it sometimes doesn't work, and it's frustrating, and it's complex. But what's really exciting to me about AI today is that it is changing the culture of computer science, and it's making it just a little bit more organic, a little bit more biological. I'm sure, raise your hands if you've used AI. I assume it's 100% of you. ChatGPT, anything. If you put the same prompt into ChatGPT 1 second after the next, you will not get the same response. And no one really understands exactly how these systems work, why they say what they say. We can sort of explain the system, but we can't perfectly predict what it's going to do. And in many ways, that's its value. It's the lack of control, it's the lack of predictability that makes these systems interesting to us. And so one of the reasons I'm really excited to kick off this panel and to talk about AI as it relates to synthetic biology is I actually think we have this really exciting opportunity to bring these cultures together rather than trying to shift the culture of biology to look like Silicon Valley or try to force computer scientists to think like biologists. And rather than sort of fighting with each other, debating one another, really finding a new language and a new way to interact with these tools and with these disciplines in a much more productive way. So I'm really excited to kick off this panel. Joining me, I have Ahir Pushpanath. He's been leading biocatalyst development efforts over the past decade plus at places like Johnson Matthey, Basecamp Research, and now he's the technical director for biocatalysis at BitBiome, which interestingly has also built a really large genomic repository from some of its proprietary sequencing technologies. And so one of the things I'm excited to talk about in this panel, and actually all three of our panelists have a really interesting perspective on this, is the importance of data in AI and the tools that we develop to generate that data and use that data. To Ahir's left, I've got Elodie Pronier. Elodie leads biomedical discovery at Oken, right here in Paris, local here, and has been a key technical contributor to the creation of BioOptimus, which is working to create a universal biological foundation model in close partnership with Oken, and brings, again, a unique multimodal data set to that from its global hospital partnerships. And finally, we have Kelly Zalukowski. She's the Senior Director of Computational Neurobiology at Recursion, one of the leaders in this field, and she leads their London office. So in this role, she oversees, among other things, Recursion's partnership with Roche Genentech, which recently, and congratulations on this, delivered their first ever licensed phenomap. And so I'm curious to talk a little bit about what the phenomap is and their largest ever financial milestone. And I mention this just because one of the things I want to cover in this panel is also the business of this. You all are still in the very lucky and privileged position of being students. I really wish I was back there. But I assume many of you want to build your careers in this field. And so there is a reality around how do we make money doing this? How do we build sustainable companies that use these tools that you've been working on, that use AI to deliver real value in the world, not just scientific value, but also commercial value? And so maybe before we get to the big picture, let's start really practical. Because I think, especially for folks in this room, they want to think about, well, what can I do today? How do I even get started? It feels overwhelming sometimes. And so I'd love to just start the discussion with where you've seen the most practical value in AI today. Where you think it's actually making a real difference, and not just the sexy, hey, we're going to go design everything from scratch, zero shot with AI, but like, where is it actually making a difference in your day-to-day? Here, we can start with you.
Yeah, thanks, Anne-Marie, for that great introduction. First of all, I want to say, honored and humbled to be part of the iGEM. We were talking previously, all the students here are doing some crazy good science, and when we were in high school, I don't think we were at that level. So it's an honor to
when some of us were in grad school, I don't think we were at this level.
Absolutely. But yeah, talking about AI, I've always felt, with any new technology that comes on the horizon, human nature is to look at the dream state. What can we do with this new technology? It's when ChatGPT, as you mentioned, when it came up, everyone's like, oh, wow, this thing can do quite a lot of stuff. And then slowly you start seeing there's limitations to the technology, and then it finds its niche. And that's the same thing that's happening with AI, I think, in also the synthetic biology space. We have a dream state where we want it to be predictive. We want it to be able to give us innovation that we can't do as humans. But I feel like it's already found a niche state where it's only accelerating or giving us advantage in the mundane activities in the synthetic biology space anyway. So it's a good data analysis tool. It's a supportive tool. It's not near an actual innovation tool at the moment. That's where I think it is. But the greatest, the hope for the AI is for it to become a predictive tool for the design of organisms and enzymes.
Yeah, and to me, there is part that's historically, AI has been already proving points in the chemistry space. I think it's the Nobel Prize recently showed us that indeed it has been accepted by the community. And to me, in biology, it is still to be proven where it can add the value. And I agree. I think the main subject is, okay, none of us, I guess, on the stage here think that AI is going to replace biologists, at least for the next few decades. And to me, it's more on supporting and accelerating what a biologist could do. And I think one good example that is becoming more and more clear is on the diagnostic. I think there is a lot of different companies that are really showing that AI can support pathologists and clinicians in their diagnosis, and especially in oncology. I think to me that's one area where currently I see a lot of impact of the AI.
And there's an entire diagnostics section over here leveraging synthetic biology, and it'll be really interesting to see over the next few years how AI starts playing into that. Kelly, where are you using it most today?
Yeah, I mean, I think there's an analogy to, this is my first time in Paris. God bless you all, I don't speak a lick of French. I'm that embarrassing American. And, I've already 5 times today pulled out my phone to say, oh goodness, how do you say this? Or to have it record and Google Translate tells me, oh, she meant this, right? And it's really effective for translating one spoken language to another spoken language, and I think in the same way, it's helping us translate in two different ways in tech bio. One of them is we create these massive data sets, so whole genome arrayed CRISPR knockout or whole transcriptome single cell sequencing knockout, and it helps us to translate that massive high-dimensional data set into something my feeble human brain can interpret, right? And the other one is, I think, a big issue that's sort of a little pain point that every tech bio company now has is like, we have biologists, we have machine learning scientists, we have data scientists, and they don't quite all speak the same language. But we're starting to develop these tools that allow our biologists and chemists to interact with these data sets directly using natural language. So instead of saying, hey, I have this kind of ill-formed question, and I'm going to go to my data scientist and frustrate them with my ill-formed question, they can say, hey, Lo, or hey, Gemini, show me the 10 most phenotypically similar genes to CDK12 and get that directly. And then they can visualize that all using natural language. And I think that's really going to accelerate that translation piece.
And I think that's also, and I'm sure the people in the room are going to be even more part of this transition, is somehow, as you said at the beginning, you have to accept as a biologist that even if not everything that you think as a biologist can be translated into a code version, is indeed making a step towards this thinking as a biologist, being like, okay, maybe there is some part of my reasoning as a biologist that actually can be transformed as a part of a code, not 100% of what I'm doing, but already if I'm getting a step there, then obviously I'm getting a step towards being able to talk and communicate the language of a computational biologist.
Yeah.
So, yeah.
I'd love to continue to pull on this thread a little bit, and Elodie and Kelly, you in particular, I think, work with both of those communities really very, very closely. And certainly at Ginkgo, for a while, and I'd say Ginkgo, was certainly leading the charge in terms of data-driven biological engineering. We were big believers in big data analysis tools to use that data. But even so, we've got a lot of really good biologists who, believe very firmly, like, none of this stuff matters unless it works in a lab, and your stuff isn't going to work in my lab. What have you found to be the best tools to start bridging that divide? You were just mentioning using some of these language models to help bridge it a bit. But I'm curious culturally what you've seen get the biologists on board and what you've seen really, I'd say, like, build respect and understanding among these groups of what the other brings and what the challenges are as you try to translate.
I think, at least to myself, I joined Oakin like 6 years ago, so I was the only biologist in the company for many years. And indeed, I did a lot of being a translator, so accepting to spend a lot of time with biologists, understanding exactly which was the easy part for me, obviously, because I'm a biologist by training. So it was, okay, discussing with clinicians, discussing with people in biology or clinics, understand what is their hope from AI, and often saying, like, okay, no, we're not there yet. Like, as we said at the beginning, like, it's not going to replace you 100%, so this is not possible from what you think from AI. This I know is possible, so let me talk then to the data scientist and explain what is your need so that you can translate it into a code. So that meant, to me, that's a lot of discussion and be a translator, being a bit of like your translator from the two team. And indeed, I think having more and more tools that help and interpret and push the understanding of the model that are interpretable by a biologist help open the door, the box, and it's not being a black box anymore. And then that helps also the communication because you have the same data that people can understand both ways.
Can I push back a little bit on this? So if tools are the way to communicate, doesn't that first have to start with ourselves as humans, that we want to communicate and be collaborative to begin with and use the tools? So it's something that I've had the luxury in my career and also at school, where my teacher told me, if you look at biology, it's actually just chemistry. And if the chemistry teacher says, actually, you know what, chemistry is just physics. So actually, everyone is looking at the other fields.
Hey, as a math major, I just want to point out physics is just math.
Well, there you go. So I think there's a human ego ego as well. In every discipline, we think that that's the major discipline. Everything else has to come via us. So we have to bridge that gap first and say, it's happening at the intersection, first of all. So how do we get this communication piece to change mentality? Is it via the tool, or is it via some other medium?
I mean, I think what we're seeing when we build these maps of biology and then we ask people to suspend disbelief a little bit and engage with what they predict, the ideal outcome for us, honestly, looks a little bit like your ideal publication, where it's like, here's a thing you know already to be true, and here's an extra novel piece. And by showing them, hey, this does put out something that makes sense for a portion of it, they're more willing to take the leap on the things that are truly novel, that they haven't seen before, that don't necessarily make sense. And so you have to help people bridge that gap between well, that's not in my benchmarking data set, that's not known ground truth, that's not, and well, but maybe it's real, right? And I think breadcrumbing them to that is kind of necessary, just because no matter what you're doing, the hardest part is still humans, right?
Yeah. That's a nice term you use, breadcrumbing, right? I think that's the way forward, spoon feeding.
Yeah. And Kelly, I know Recursion has obviously built a very large automated lab. I suspect part of the way you breadcrumb people along is you also do a lot of experimentation to prove that this stuff is actually driving value over time. As a framework for folks, I'm sure you've heard this before. When I think about what drives value in AI, there's compute, which is what most people talk about, right? Bigger models, more powerful, yada, yada, yada. That was sort of the ChatGPT moment. There's technology, so new architectures, new algorithms. You could also call that talent, right? People who know how to build and use these systems. And then there's data, right? What is the data that we're able to train these models on? And there's sort of a reductionist view that AI is only as good as the data, and it's learning relationships between that data, but it really can't generalize beyond that. That's a debatable topic, just to be clear. But there's no question that data is key to building and scaling these models. And for a long time, I'd say in biology that has certainly been the biggest bottleneck and likely will continue to be the biggest bottleneck for some time. And so I'd like to have a conversation on the role of data, and maybe specifically how you see the role of data both on the pre-training side of AI models, so just building, maybe Elodie in the case of BioOptimist, building a foundation model for biology. What does that mean? What is the data you're going to use? When will enough be enough? And then also on the sort of fine-tuning, reinforcement learning, validation side, how you see the role of data, kind of throughout the life cycle of AI. Kelly, if you want to start, just given Recursion's role.
Yeah, definitely. I think Recursion's point of view is a key differentiator that Recursion has is that we are building these scaled, fit-for-purpose, high-quality, consistent data sets in-house. And so the main technology that Recursion started with 10 years ago was cell painting. We do a massive amount of high-content imaging. And honestly, the first half-decade of Recursion's existence was doing all of these, call them boring things, but it was like the control strategy, the standardization, the automation with all of the robotics in the lab. How do you guarantee that an image we took in 2015 we can directly compare to an image we will take in 2025? Right? That's anyone who's worked in a lab knows that that's a very, very hard thing to do, because often you can't compare an image you took on Monday to an image you took on Wednesday, right? So a 10-year gap is really something. So I think really putting in the effort in the ground stage to create the data you need to meaningfully deploy AI against biology is one thing. And then to connect it to our previous conversation, if we get a prediction out of our maps of biology, the very next thing we do is say, oh, okay, how can we orthogonally validate that? What is the next assay we can use to help prove out that, hey, that thing we're seeing in our map is likely to be true, or we're convincing ourselves and we're convincing our partners. And a big part of the Recursion philosophy on doing this is that we're trying to collect all of these high-dimensional data sets in a way that they can be fed back into the beginning. So we create these virtuous cycles. So any data point we collect for a specific program feeds back into the original training data set, and it's available to every future program we will generate. And I think that is going to help. That closed lab in the loop piece is a key part of the philosophy of Recursion as well.
And I think it's interesting because at Okin we took exactly the opposite vision, which comes with the same limitation, but on the other side. So the way we are building the foundation model is because we think that we want to treat humans. So the best way to treat human is to find human data. So indeed, we don't have a lot of data in-house currently, but we're looking and working a lot with academic partners, people from clinicians, people in the real world to get access to their data, and that's how we build the models. Like having a lot of different type of data, so multimodalities, on a lot of different patients, so that we hope, and again, it's a hope, it's being tested currently, that you have less bias in the way you develop your models because they have seen more type of patient and more type of data. But then so that, on principle, is amazing because then you force the model not to be so overfitting in one population. But then you have to validate the results of your finding, and then you go back to a lab and you go back to a petri dish, which you know is not representative of your population of interest from the beginning. And then what do? If you validate your finding, then happy ending, happy story. If you don't validate, who is wrong? Is it the AI or is it your petri dish? And right now, it's really, really a complex question for us.
Yeah, and I think you're both operating in this incredibly kind of difficult field where we just don't have models, whether you're talking about a computational model or an animal model, that are reliably translatable to whatever that next step is, right? The computational model is so-so when you get to a petri dish, which is so-so when you get to an animal, which is so-so when you get to a human, and so you have all these different translation steps that you're trying to bridge. Is it easier in biocatalysis?
You know, I was just going to say, maybe it's a little easier because we don't have to do with mouse and things like that. But I think the lab scale results you get with an industrial enzyme is very different to the enzyme performance in a reactor at process scale. So we do still have that same problem with data. If you're going to find data, do you use lab data to train all your models, or do you use process data from actual plants, right? So I agree with both of you in the way that lab in the loop is a must also for industrial biotechnology. If you're going to be developing any enzymes that work in a process, you've got to validate it at some point. And data for me is not just for AI, but data is the only thing the customers trust ultimately. They're not going to trust your hypothesis on AI. They're going to be trusting, does it work, right? Your final product of AI. So for me, I'm always looking at data. Can we get the right data, which the customer is convinced with? Is that the right data then to train the AI model so we can give more solutions to customers? That's how I'm viewing the whole data aspect in AI.
How do you all think about, you just mentioned data is the only thing, lab data is the only thing that customers trust. I think when folks talk about AI in biotech and SynBio, they're typically focused on the discovery side, at least this type of community is. So we're going to discover a novel target, we're going to discover a novel biomarker, we're going to discover a novel therapeutic. And some of the pushback I've heard recently from the broader community is, yeah, but that's not actually the problem. Hypothesis generation isn't the problem. Hypothesis testing is a problem, and what hypothesis testing really means is you stick it in a giant bioreactor. You stick it in a human in a clinical trial, and that is the bottleneck. And so I'm curious how you see the space evolving as we get a lot better at hypothesis generation, and I think that's already happening. I'm sure, at Recursion you've got 100x, 1,000x more great ideas than you can actually test in a lab, much less a clinical trial. And so how are you thinking about the ecosystem evolution, given that we have this massive influx of design and hypothesis, and we're still bottlenecked at that very last stage?
Yeah, that's a very good one. I think that's exactly where automation comes into play as well. So the way you collect data has to be at the same pace that AI is craving the data. So this is a really important thing for the lab in the loop. The way you collect the data has to be really, really fast-paced. It has to be cheap as well, and the types of data you collect needs to be reflective of the customer problem. So those, I feel, are the major limitations of data collection in general for AI. And if the tools and the engineering principles don't catch up and automation doesn't catch up, AI is always going to be in the state of, we need better data, we need better data. So that's where I'm at.
But to me, one of the main limitations of this is obviously going to be money.
Yeah.
Because if in an ideal world, I would love AI to create indeed thousands of hypotheses a year, a second, a minute, you tell me, and then to test them in the lab. So I guess indeed automation is one step, but I don't know if in this room or ever I would...
Anyone have money?
You have billions of euros to spend on this? Because to me that's the dream I have, which is, okay, I have thousands of hypotheses, I have hundreds of models on which I can test my hypothesis and not rely only on one model. And then I will have the ground truth that we are all looking about. But when I'm thinking of this, then who does the check? I haven't found that person yet, so.
Yeah, I mean, one thing is, I think you're totally right. Like, it's thinking about how do you miniaturize, automate, industrialize, scale that next step always. And so, like, recursion always starts with high content imaging, but like we've also collected more than 1 million transcriptomes, right? In 1536 well plates, compounds, like miniaturizing that process has helped us to collect data at scale on the next validation step. But I think another way we can think about it is like AI can help point us to the most fruitful places to do that testing. Like, can we intelligently deploy active learning to say, oh, well, what is the chemistry library we should create to most efficiently explore the space, given that there's more possible chemical structures than there are stars in the universe, right? We can't possibly scan them all. Like, can AI point us in the direction of like, okay, this is where you would start, and then this is the most fruitful direction, and to do that sort of like binary search more quickly, I think is another place where it's going to help speed up that development process.
You all are talking a lot about data generation upfront as part of your process, validation. How have you balanced the different types of data you could generate? There's data you generate for a customer program, right? Hey, I need to fine-tune my model in this particular class of enzymes, or I need to go do a bunch of validation work, versus I think my model is hungry for this type of data. Like, I think it'll just get better. I don't really know exactly how or why, but it seems hungry for that. Like, how are you thinking about what I would maybe describe as like proactive data generation and more generalized data generation, or more, maybe you could call it like unbiased, versus focused on an individual program?
Yeah, I think there's a big hypothesis in the field of enzyme design anyway, that if you really have the biggest data set possible of natural enzymes, then AI is somehow going to learn the ground truth of what it means to be a functional natural enzyme. So that kind of data is more not led by the lab, but it's for generative AI, surely. So here, collecting more natural data is what BitBiome does, and we've built this huge 2 billion sequence database of microbial diversity. And the hope there is maybe we feed it enough diversity, AI will finally find the ground truth of what it means to make a natural enzyme. But is that going to be enough then to make an industrial enzyme? That one is not represented in natural enzymes. So you have to have the lab in the loop for that kind of data. So both is necessary, I feel. You need to have that vision of let's just feed it more data, and at some point you'll get some answer. And you also need, I'm going to kind of navigate it to this particular process-ready enzyme state. So that's where I am on that. Yeah.
I think so at Recursion, a thing that we're doing, my first year at Recursion, we all got a box of gifts for Christmas, and one of them was the book Loon Shots, which is sort of centered on the idea that like ideas that are worth chasing, at the beginning people are going to say, you're crazy, that's never going to work, and then some sort of process ways to go about like, how do you do this sustainably, chasing loon shots? And one of the key themes in the book was like, okay, you're going to meaningfully wall off in your company the artists from the soldiers. And so we have a whole department at Recursion, actually a large fraction of the company, that's dedicated to building and maintaining the Recursion operating system, the Recursion OS, which is like proactively generating more maps in new cell types, proactively developing more powerful models to just like keep building what we're doing at Recursion to serve all of our partnerships and all of our programs. And then we sort of wall off our machine learning researchers or our people who are looking at these like longer term, this isn't going to read out for 3 years or 5 years, but if it works, it will be game changing. And so we sort of operationally make sure that that happens.
Well, us, it's a bit of more a testing and see if we're happy kind of approach, meaning that obviously a lot of data scientists that are coming at Oakin, they are really good at using images as a data modality to make any predictions. So obviously we have, and even on Bioptimus, the foundation model, they are working amazingly well on image-based prediction. So that's an easy avenue that was for us to take. And then on the omics data, to me, it's not as striking as I said at the beginning, like where AI is going to be able to change to bring the gap or to change something. And so we think that the more the merrier, because obviously then you can grasp better the heterogeneity of the disease. Is that going to be the best suitable approach for AI? I'll come back in 2 years and I'll tell you.
I like this incremental approach. Like one of the questions I get asked a lot is like, okay, but like how much data do you need? And you mentioned money earlier. One of the groups I work with is DARPA, the United States kind of Defense Department's research group. And they want to know this question too. Like they've got money. They're willing to spend the money. They want to know, what should we spend it on? Like how do we know that if we go put hundreds of millions, billions of dollars to work to generate the data that you all in the community want, how do we know that's going to make a difference? How do we know that's going to be enough? And so you've mentioned kind of these little hypotheses that you're testing. How do you all start getting conviction around, hey, I think investing in this area of data generation, because the reality is if data generation is the bottleneck, data generation is also really expensive. And you have to therefore have real conviction before you spend limited resources on that. How do you build that conviction, and how would you help the people who do have the money, build the conviction to start putting it where you want it?
So us, we have done a lot of testing on because obviously people always tell you what is the minimum that you need, because it's always better to start with the minimum. So we did a lot of testing on every not every, but the majority of the predictive models that we have at Oaken and by Optimus, we first kind of synthetic, like in a computer, you can kind of see the impact of the numbers on the performances of your model and say okay, after this bar, I know that the learning and the performances of the model are really bad, so that's the minimum that we usually require. And obviously you can do that by modality, or even if you have different approaches and type of data to kind of say what is your minimum. And then there's amazing people that indeed, even when they have complex model, they are trying to also see what was the weight of every independent modality in the performance of the model. So if you lose one modality, what is the impact on the performance of the model? So I'm not saying I have an answer that is always clear when one customer is coming to me okay, I want to predict patient survival. How many data and what type of data do I need? I don't have a clear answer, but I start to have an idea of what is at least the minimum required. Then, is the minimum sufficient, or do I want thousands more? It's still a debate for me.
I have two things to say about this. One of them is at Recursion, I think we find we have this milk and cookies problem, which is you finish your cookie, you still have a little more milk. Oh, well, I guess I need another cookie. Oh, no, now I need more milk. Oh, no, I need... And what we're seeing is we get to be really actually kind of data rich and then compute limited. And then we invest very heavily in compute. So recently, I think over the summer, we rolled out what is now the largest in biopharma compute cluster. And by deploying our newest foundation model on that compute cluster, we improved recall in our benchmarks by 25% in 2 weeks. So clearly we were compute limited, right? And now we're back to saying, okay, well, now we have enough compute. What data should we generate? But I think the other place we're really thinking a lot is around benchmarking and what does good look like in this space, given that you're trying to discover the unknown and find novel things. And so Valence Labs at Recursion, which is this machine learning research arm, has recently started this collaboration. I think on the initial publication we're J&J, Bayer, AstraZeneca, and Recursion, thinking about okay, how can we all come together to say these are going to be the benchmarking data sets. These are going to be the sort of ground truths. This is the MNIST data set for TechBio, right? When we're testing these models together, can we all sort of come to a consensus around how we will test them and how we will decide what good looks like? Then we can say oh, how are we doing against these benchmarks we all understand?
I think, Anne-Marie, if the question is more around how do we get the investors to put money into getting these data sets, I think it's already happening. A lot of startups are still raising pretty substantial amounts, even in this bad climate of investors. So they're managing to convince the people with money that there is a promise of AI. Yep. But are they convincing them about exactly the data sets required to pull it off? No. So that's the issue. I think when you get to the honesty, I don't think a lot of us, even scientists, we have to be a bit humble here. Do we really understand biology enough to know exactly what data set to collect? So you're making a gamble there, first of all, on what kind of data set we need, right? It is. And then once you've gambled, and you've taken that leap, you bring all the millions into it, and in halfway you find it's not working, then that's on you, right? So that fear is there for the people asking for the money as well, so we don't talk about it. We talk about only the promise and premise. So I think there's a thing that we can learn about asking for money from people that do have it. Maybe we just need to be a bit more honest at the beginning, so they give that minimum, and they don't give too much, and then you're like scrambling, trying to make up the profit. So that could be it. Yeah.
Yeah, and to me, because there is days, I'm not going to lie, at Okin, where we have amazing data and we're trying AI, and it fails. That's the reality, right? But I'm always thinking that, okay, even if you don't do fancy AI, we have data that was not existing before, and you do computational biology, and you find other answers that are not for AI, but are more for even a biologist like me, like more ground finding. And to me, that's still like a really important step towards understanding the biology, understanding the disease that we're working on. So I agree. Sometimes I'm like, yeah, we're gonna go skyrocket with AI, and I'm like, okay, it doesn't work. Okay, still not useless because we can use those data for something.
Spoken like a true scientist.
Trying to be.
Although I will put in a small plug for a topic I've gotten really interested in, which is, and there's sort of, some of what you've said, Kelly, there's kind of the unsexy blocking and tackling side of it, and then there's the really exciting kind of autonomous science vision. And one of the really interesting foundational research problems in that is, can a model develop the experiment to test its hypothesis? And if one of those hypotheses or one of those goals of the model itself is, I want to get a lot better at developing hypotheses in a particular area, then you start having a partner in these models that can help you determine where to go generate data. And I think that'll be really exciting. We've got just under 10 minutes left, and I want to chat a little bit about the business model. And we've started talking about money and investors, and so we're sort of naturally getting into this space. I think we've already seen, and this is certainly true in pharma. I think it's probably also true in industrial biotech to some degree, but it's very stark in pharma, where you used to see these fully vertically integrated pharmaceutical companies that did everything from drug design to development to distribution and marketing. And so they owned that entire value chain. And now, well over half of drugs that go into a pharmaceutical company's pipeline is partnered or acquired from a smaller, more innovative biotech. And obviously, Recursion's business model is doing a lot of that development, such that can then partner with pharmaceutical companies. And so, in many ways, the role of pharma has gotten squeezed into this much smaller piece around late-stage development and then distribution and marketing. As AI, comes more into the fold on discovery, is there a risk that kind of more traditional biotech gets squeezed in the middle, where we're not doing, distribution and commercialization, and now suddenly one-person companies run by the folks in this room in high school can do really amazing design work? Are we stuck in that translation phase, or how do you think the business model evolves in this field as AI develops?
It's a tough question. I think in the pharma space anyway, it would be a great reality when anyone in this room could make their own multi-billion-dollar pharma. That's what I want to see. Why is it only a couple of 15, 20 handful of pharmas running the game, when really AI has empowered all of us to essentially become our own innovator and own distributor? But it's a hard game, right? So that's the thing. You need the money, as we go to before, and you need to have the proof points. But the way I see it, the business models for the small fish at the moment is still reliant on the big players to take a gamble, and then at some point the big players buy them out. Or you take the risk and say, I believe so much that I can go to market with my own therapeutic. I don't need a big player to support me here. But that requires your investors to take the risk with you. So that, I feel, is a gamble again at the very beginning of what kind of model you want to follow. And at some point you're going to have half-half. You're going to have to be in bed with one of the big players as well.
Yeah, I mean, at Recursion, we have a number of different irons in the fire. I think our main thing that everyone knows us for is developing drugs and developing drug discovery programs and partnering those out with large partners. But we're also doing things like we are partnered with NVIDIA, and we've put some of our models on BioNeMo so that other people, even you guys, could access those models and work with them. Or Another thing we're doing is taking the large language model tools I was talking about earlier. One of them we built internally is called Low. And we are using sort of an app store model where you're like creating the ecosystem and you have the content. And so Bayer is the first company that's working with Low, but we're allowing them to use these large language model enabled tools to directly query recursion data. And so it's like, oh, you want to get insights into recursion data? Well, here's the app you're going to use to do that. And hopefully by that, you're sort of capturing some of that space.
And to me, it's really complex because, I think I've seen a bit all, because Bioptimus is obviously a tech company, so we are more talking about delivering a platform that is indeed hopefully delivering to the world really good algorithm. Is that enough as a business? I don't know. I think right now it is indeed. Because AI is the hype, it's really interesting to investors. But then when you pass the phase of investor, what happened to us? I don't know. Which was the beginning of Okin as well, where we started really as a tech. And then you go to the big players, like the happy few, that has the money to kind of bet on your tech and your platform. But then either your tech is working really well and indeed they buy yourself, so which means that it's a bit the end of your own story, or they are I like your idea and I'm going to try to do it myself, which is obviously then their bet to kind of good luck to kind of replace a company like us that are really focused on developing AI. Or you take the bet of why can't I become myself one of the big players that I'm founding my own targets and I go to the drug development. But that's then balancing the investment that is coming from tech people that are the ones that usually invest in you at the beginning versus having enough money to fight for the 15 years that is going to take you to have the proof point that you have an asset that indeed was successful in clinical trial. To me, there is a bubble in between that is really the challenge to a lot of companies like us for the moment.
I mean, supposing any of our technologies we're developing suddenly becomes, and you have a eureka moment and oh my God, this is actually working, will you take the gamble then and say, you know what, I'm going to make my own thing. I'm going to be making products with my technology rather than selling it on? Is that what we're all waiting for, to have that breakthrough moment?
Well, I think I think this actually reflects a little bit of the bias in our industry, where right now the only way to prove that your technology has any value is to make an asset that is valuable with it.
Yeah.
And the unfortunate side effect of that model is that there are very few, if any, businesses in our space that have sustained R&D, have sustained innovation after they made a product.
Yeah.
Either the product fails and everyone's like, oh, well, the technology must have been garbage because the product didn't work, or the asset is wildly successful, gets acquired by pharma, and the R&D team still doesn't have a job to do, because now the only thing anyone cares about is that drug, because that's where all the value is. And I'll say, for me, one of the things I'm actually most excited about and optimistic about is the potential of AI to allow biotech, tech bio, SynBio, whatever we want to call it, companies to monetize the R&D process itself, so that innovation engine can in itself be a sustained business model and isn't only reliant on those assets. Obviously, the assets will still be maybe the most valuable piece, but is there a way to at least sustain that innovation engine because AI can help make some of this technology more translatable? We only have a couple minutes left. I want to be cognizant of our audience here. These are all college students and even high school students who are building their studies and probably their careers in this space. I'd love to just go around and get a 30-second kind of piece of advice from you on for this group.
Yeah, I mean, when I started grad school at Stanford, I was coming from having done college in Arkansas, which is a place that's not particularly known for being really tech forward, and I landed at Stanford and I was like, I don't understand why anyone would do computer science. My roommate was a math major, and when she said for loop for the first time, I imagined a clover leaf. No idea. None. But once you see that oh, that's the water we swim in, that's what you're going to need in order to be successful here, I went and started learning those things, and now, this is what I'm doing. I'm a major leader at a leading tech bio company with on the bleeding edge of things. And so like, I think my call would say like, don't be intimidated by what you don't know. Like a lot of these things are really less difficult than you make them out to be in the beginning when you know nothing and it seems intimidating. Just getting that first step in lets you know that you can do all of these things.
It's also believe on thinking outside of the box is really important, because I already knew I wanted to do biology, but you know, so I did a PhD and then I did a postdoc and then I wanted to have my own lab and blah blah blah. You know, this kind of like academic career path that everyone tells you when you're starting. I mean, when I started at the time, and when I joined Okin, I was like, again, it was a small company of 40 people. I was the only biologist full of like data scientists that I remember the first lab meeting I went to, it was full of mathematic function and I was like, whoa, whoa, that's a huge gap from being a lab rat, right? And so to me at the first I was like, what bad can happen? Like, I'm going to have an experience, and if it fails, I'm going to still open the door to something that I was not thinking I was capable of. And here I am 6 years after. Now I understand what AI is, I understand the math function, and I'm even able to talk about it. So I think, yeah, trust in yourself and saying like, okay, it's not exactly the path that was supposed to be mine, but let's see where it goes.
Yeah, I think both of those are very good. I'll just add that we're living in the information age. When we were in school, and I guess information was there, but not at the level it is at the moment. So as you in high school and undergrad, PhDs, you're all fully saturated with information, and there's so many worldwide issues that are happening. You feel stressed that you have to be, we are relying on you to solve the next problems of the world. So that pressure sometimes can distract you from doing the scientific rigor that's needed to achieve. So I just follow your heart and go one step at a time. Don't get over-pressured into delivering the future solutions.
Yeah. Thank you all so much for joining. The only thing I would add is become bilingual or trilingual. Like, learn computer science, learn biology, learn business so that you can help be a translator, just as all these folks have become translators in their industry. Congratulations again. I'm so proud of all of you, and thanks so much for sticking with us. Have a good one.
Thank you.